Today, I will be presenting the four fundamental molecules of biology. The first compound is carbohydrate. Monosaccharides are the monomers from which carbohydrates are made. Here is the general formula for the structure of monosaccharides. A simple hexose sugar known as glucose is the most basic monomer with the chemical formula of C6H12O6. The isomers of glucose make up the monomers of carbohydrates. The two isomers you need to be aware of are fructose, found in plants that acts as a transport sugar, and galactose, found in mammalian milk. These contain the same chemical formula but have a different structure as shown here. Note that fructose is a pentose sugar, and we will explore other sugar structures when discussing nucleic acids. I will also show to you the differences between alpha and beta type of glucose depending on the position of the hydroxyl groups in the molecule. Now, disaccharides, which are combinations of two sugar molecules, also play a role in the composition of carbohydrates. And the most important one is maltose, as shown here, which is formed by the digestion of starch by amylase and found in germinating seeds. In order to form maltose, two molecules of glucose undergo a condensation reaction, resulting in a glycosidic bond and the release of a water molecule. Hydrolysis reactions are the opposite of this, breaking down polymers into monomers using water. The polymer formed from these monomers is known as a polysaccharide. A strong example is starch, and a particular type known as amylopectin is shown here. Starch is an essential compound in nature and is found in cellulose. Benedict's test is used to test for sugars. Lipids are hydrophobic compounds and are not polymers. The most common lipids are triglycerides and phospholipids. Here is their general formula. Triglycerides, as shown here, are used for storage, insulation, and protection of organs. They yield more energy per unit mass than carbohydrates, but cannot be mobilized quickly since they are insoluble. Three condensation reactions, as shown here, are needed to form triglyceride with the use of ester bonding. The structure of a lipid is composed of glycerol and fatty acids. Glycerol is shown here, and fatty acids complex structure is also shown here, consisting or containing hydrocarbon chains and a carboxyl group. The difference between saturated and unsaturated fatty acid is that along the long chain of the fatty acid molecule, there are in unsaturated fatty acids double bonds which make the molecule of the fatty acid assume a non-straight structure, rather have a kinked shape, as we say, which when embedded in membrane structures, they impart more of fluidity in the membrane and more easily to move the molecules of cholesterol in and out of membrane. Phospholipids are the main components of cell membranes. A phosphate group exists in place of a fatty acid chain in their structure, characterized by a polar hydrophilic head, phosphate group, and two nonpolar hydrophobic tails. When mixed with water, they form liposomes. Emulsion test can be used to test lipid. Proteins are used in our body structure as enzymes for transport, as hormones, as antibodies, and for storage. Every structure, cellular, tissue, or organ function is mediated by some kind of protein. Proteins therefore used in our body structure and function as enzymes for transport processes, as hormones and signals, as antibodies, communication between cells or intracellular paths of signaling, and for storage and other functions. Here's the general formula. The proteins are made out of a sequence of amino acids, which forms the primary structure of a protein, and different foldings or changes in the direction of the primary structure in space and result in secondary structures such as helices, beta-pleated sheets, or motifs other protein structures, and the tertiary structure where the protein holds in space in three-dimensional space. Finally, a quaternary structure, and you combine different peptides to more complex compound conglomerates, such as the heme ring, hemoglobin, made out of 4 protein polypeptides, alpha and 2 beta globins, structure protein, a heme ring with an extra moiety attached to the protein that gives special functional. For the iron in the heme ring, it chemically binds oxygen. Proteins, therefore, are made out of building blocks of the proteins, or monomers of the proteins are the amino acids. Amino acids contain central carbon, which an amino group, a carboxyl group attached. Also a third variable group, alpha carbon positions, we say, shown here. Animal organisms, there are 20 different amino acids that all proteins are made of. Same ones hold for humans. 2 or multiple amino acids join. Dipeptide or a polypeptide is formed. Proteins or structural components. Primary structure is a sequence of amino acids in a polypeptide chain. Secondary structure made out of hydrogen bonds between carboxyl groups and amino groups, which shape the protein structure in a two-dimensional plane. Usually, tertiary structure is held together by bonds between variable groups. For example, the disulfide bridges between cysteine amino acids create strong foldings in space, in the three-dimensional space, account for the tertiary structure of proteins. Quaternary structure. The protein is composed of different polypeptide chains, other organic or inorganic moieties. Proteins are globular, fibrous proteins, functional proteins. They have globular structures, while fibrous proteins are of a structural function. Enzymes are essential compounds, again, a significant role in catalyzing reactions. Not actually change energy. The starting reacting and reacting compounds and the products not change the energy levels. What they actually do is that they speed up process called catalysis. Fourth group, nucleic acids made up of monomers called nucleotides, which have this overall structure. DNA contains structure shown here. The presence of a pentose sugar, in the case of DNA, is deoxyribose. In the case of RNA, ribose, providing properties to RNA, DNA, and RNA molecules respectively. Two nucleic acids have some key differences. Also, a messenger RNA, mRNA, transfer RNA, tRNA, will be for another video. Monomers of nucleic acids, nucleotides, rise by forming phosphodiester bonds, phosphodiester bonds in carbon 3 on pentose, oxygen from phosphate group, also be dinucleotides and polynucleotides. Nucleotides have 5' end attached to 5 of the pentose sugar and a 3' end hydroxy group attached to carbon 3 of the pentose sugar. Here, replication function and ATP formation are all important in organisms covered in another video presentation together. Stay tuned to come up. Your attention.